Ever feel like your body is this incredibly sophisticated, perfectly tuned power plant, constantly adjusting its energy output, its fuel intake, all without you thinking about it?
It really is remarkable when you stop to consider it.
Or imagine trying to power an entire sprawling city without a central energy grid or a fuel delivery system. That's kind of the monumental task our bodies pull off second by second with amazing efficiency.
That's a great analogy, actually. It
Yeah.
captures the complexity and the constant need for regulation.
Today, we're taking a deep dive into the very core of this biological marvel. We're focusing on Section 4 of Mark's Basic Medical Biochemistry, specifically looking at carbohydrate metabolism, fuel oxidation, and the generation of adenosine triphosphate, ATP.
Hmm.
And alongside that, how hormones regulate it, and how carbs make their way through our digestive system.
It's a foundational piece of biochemistry, really tying diet to cellular function.
So our mission really is to guide you through these complex pathways, help you connect the dots between, say, your lunch and your actual cellular energy. We want to give you those clear aha moments, those practical insights into how your body really works without getting bogged down in, well, too much jargon.
Absolutely. I mean, we'll be drawing insights from across this entire section, showing how it all fits together. We start with the fundamentals, carb metabolism, energy generation. Then we'll look at the specific roles of hormones, the body's master regulators, if you will.
The conductors of the orchestra.
Exactly. And finally, trace that path of carbohydrates right from your plate, through digestion, and into your cells. It's a truly holistic view, step by step, of how we power ourselves, really emphasizing how interconnected these systems are. The core: carbohydrate metabolism, fuel oxidation, and ATP generation.
Okay, let's unpack this incredible system then. When we talk about fuel for the body, especially carbohydrates, what's the superstar molecule, the absolute king of energy, and why is it so central to, well, everything?
Well, what's truly fascinating here, and maybe one of the biggest aha moments in biochemistry, is that glucose isn't just a fuel. It's the universal fuel.
Universal, meaning every single cell uses it.
Every single human cell, without exception, utilizes glucose to obtain energy. Think of it as the most versatile, readily accepted currency in your body. Your brain cells, for example, are almost exclusively reliant on it. Muscle cells need it for bursts of activity. But the story doesn't stop there. Glucose is also a primary source of carbon atoms. It's a foundational building block for synthesizing a huge range of other essential compounds.
Ah, so it's not just burned for energy, it's used to build things too.
Precisely. Imagine it not just as the gasoline powering a sophisticated car factory, but also as the fundamental steel, plastic, and rubber needed to build every part of that car. Its versatility is just profound.
Okay, like what sort of things? Give us some examples.
Sure. For instance, glucose is a precursor for other sugars, like lactose, the sugar found in milk, which is synthesized in mammary glands. It also forms crucial components for cell surface antigens, vital for cell recognition, immune responses. It's used to build nucleotides, the very building blocks of your DNA and RNA.
Wow, okay. DNA building blocks from sugar.
Yep. And glycosaminoglycans, these are large, complex sugar molecules essential for the extracellular matrix and cartilage. They provide structural support, lubrication, shock absorption, all throughout your body. And beyond those roles, glucose can be converted into lipids, including fatty acids, cholesterol, various steroid hormones, which have roles from energy storage to cell communication.
So hormones too.
Certain cell hormones, yes. It can also form nucleic acids, and even many of the non-essential amino acids. It just highlights how central glucose is. The only major things our bodies cannot synthesize from glucose are vitamins, essential amino acids, and essential fatty acids, those we have to get from our diet.
That's incredible adaptability, using the same molecule for fuel and construction material.
Exactly. It speaks volumes about evolutionary efficiency.
So glucose truly is king, doing double duty. But our diet isn't just pure glucose, right? We eat other sugars like fructose from fruit, galactose from milk, lots of complex starches. Do they just get tossed aside, or do they somehow join this glucose party?
Oh, they absolutely join the party. They don't get discarded at all. And often, they get converted pretty quickly into forms that fit right into glucose's metabolic pathways. If we connect this to the bigger picture, all these dietary monosaccharides, glucose, galactose, and fructose, they're absorbed from your digestive tract into the bloodstream. Once they get inside your tissues, they're metabolized, often converting to intermediates of glucose metabolism. So they all eventually feed into the main glucose pathways.
Okay, so they merge onto the main highway.
Exactly. Think of it like different rivers, fructose, galactose, flowing into one major metabolic delta, which is this vast interconnected network of glucose pathways. Once glucose is absorbed and gets inside a cell, it's immediately phosphorylated. A phosphate group is added by an enzyme called hexokinase.
Phosphorylated. Why?
That step does two crucial things. It traps the glucose inside the cell because the phosphorylated form can't easily cross the membrane back out, and it creates glucose 6-phosphate. And this glucose 6-phosphate molecule is, well, it's a major crossroads, a central metabolic hub.
A molecular traffic circle.
Yeah, that's a good way to put it. It's the branching point for several critical metabolic pathways. If you could visualize figures Value 1.2 and IV 0.3 from the source, they show these branching roads.
Okay, so what are the main roads leading out of this glucose 6-phosphate circle?
Well, one of the most important is glycolysis. This is the primary pathway for oxidizing glucose 6-phosphate, and it's absolutely crucial for producing ATP, the cell's immediate energy currency. Glycolysis itself actually has two modes depending on oxygen availability. There's anaerobic glycolysis.
Anaerobic, meaning without oxygen.
Exactly. This happens in cells that lack mitochondria, like your red blood cells, or under conditions of low oxygen, think intense, short bursts of muscle activity, like sprinting. Here, glucose gets converted to pyruvate and then further to lactate. It generates a small but very rapid burst of ATP. It's quick, but not very efficient, like the body's emergency power generator.
Got it. Quick and dirty energy.
Sort of, yeah. Then there's aerobic glycolysis. This happens in cells with mitochondria when oxygen is available. Here, the pyruvate from glycolysis gets fully oxidized to carbon dioxide and water. This happens through what we call the tricarboxylic acid, TCA cycle, and the electron transport chain, ETC. These pathways generate a significantly larger amount of ATP.
Right, the TCA cycle and ETC, those are the big energy producers.
They are. Think of the TCA cycle as the main combustion chamber where the fuel gets completely burnt to release maximum energy. And the ETC is like the powerful turbine that converts that released energy into the vast majority of our ATP. It's the most efficient system by far.
Okay, so glycolysis for energy, aerobic or anaerobic. What else can glucose 6-phosphate become?
Another important fate is the pentose phosphate pathway. Figure 5e.6 in the text illustrates this one. This pathway isn't primarily about bulk ATP production. It has two other main crucial functions.
Okay, what are those?
First, it generates NADPH. This is a different kind of reduced coenzyme than the NADH from glycolysis. NADPH is vital for many biosynthetic reactions, building things up like synthesizing fatty acids and cholesterol. It's also crucial for protecting cells from oxidative damage. It acts as a cellular antioxidant, helping to neutralize harmful reactive oxygen species.
So it protects the cell while also providing building blocks.
Precisely. Second, the pentose phosphate pathway produces pentose sugars. These are 5 carbon sugars, and they're absolutely vital for nucleotide synthesis, making the building blocks of DNA and RNA. And this pathway is pretty flexible. It has both oxidative and non-oxidative reactions that allow the cell to interconvert 6 carbon sugars, like glucose derivatives, and 5 carbon sugars as needed.
Flexible and essential. Okay, what's the third major route for glucose 6-phosphate?
The third major route is storage. Glucose 6-phosphate can be converted to UDP glucose, and UDP glucose is the direct precursor used to synthesize glycogen.
Glycogen, that's our stored glucose, right? Like a sugar savings account.
Exactly. It's the body's primary storage polymer for glucose, our short-term carbohydrate savings account. The largest stores are found in our muscles and liver. Muscle glycogen provides an immediate energy source for muscle contraction. Think lifting weights or running. Liver glycogen is crucial for maintaining stable blood glucose levels, especially between meals or during fasting. It ensures your brain, which needs a constant glucose supply, always has fuel.
So muscle glycogen is for the muscle itself, liver glycogen is for the whole body.
That's a great summary, yes. And that UDP glucose precursor, it's actually quite versatile itself. Figure IV.7 shows its diverse products. It's not just for glycogen, it's also involved in forming UDP galactose, which is essential for making lactose in the mammary gland during lactation.
For milk production.
Right. And it forms UDP glucuronate. This molecule is vital for detoxification. It's used to form glucuronides, which are water-soluble conjugates that help the body excrete toxic compounds like bilirubin, a breakdown product of red blood cells and many drugs.
So it helps us get rid of waste and toxins.
It does. Furthermore, nucleotide sugars derived from UDP glucose are used for synthesizing glycoproteins and glycolipids, crucial components of our extracellular matrix, cartilage, and cell membranes.
Wow. So glucose 6-phosphate really is a central hub, directing traffic to energy production, antioxidant defense, DNA building blocks, storage, and even detoxification.
It's the ultimate metabolic chameleon, absolutely central to carbohydrate metabolism.
That's an incredible web of pathways, a lot of different directions for glucose to go.
So let's circle back to energy. What's main currency here? The molecule that actually funds all this activity and makes the cellular machinery go.
That's the crucial question, isn't it? How does the body use all this potential energy? The central molecule, the universal energy currency, is adenosine triphosphate, ATP.
ATP, the famous one. The famous one. Its high-energy phosphate bonds are literally the universal energy currency of the cell. And the aha here is that it's not just energy, it's rechargeable energy.
Rechargeable, like a battery.
Exactly like a rechargeable battery. Think of ATP as the readily spendable cash in your wallet. It's constantly being spent, hydrolyzed to ADP to power things, and then immediately replenished, ADP phosphorylated back to ATP, using the energy released from oxidizing fuels like glucose. Every single physiological process that requires energy muscle contraction, moving molecules across membranes via active transport, building new proteins or DNA involves the spending of ATP.
So maintaining ATP levels must be critical.
Absolutely critical.
Yeah.
Which leads us to glucose homeostasis. Figure IV.8 in the text really depicts this well. Maintaining blood glucose within a very narrow range, roughly 70 to 100 milligrams per deciliter, is essential for survival, especially for your brain.
Why, especially the brain?
Because the brain primarily uses glucose for fuel, and, importantly, it can't store much glycogen. So it needs a constant, steady supply from the blood. This tight regulation is our body's continuous balancing act. If blood glucose levels drop too low, that's hypoglycemia, the body counters it quickly. First, by breaking down liver glycogen through glycogenolysis.
Releasing stored glucose from the liver.
Right. If that's not enough, or if glycogen stores are low, the liver starts making new glucose from non-carbohydrate sources like lactate, glycerol from fats, and amino acids. That process is called gluconeogenesis.
Making new glucose from scratch, essentially.
Pretty much. Conversely, if blood glucose gets too high, hyperglycemia, maybe after a carb-rich meal, the body shifts gears to store the excess. It converts the extra glucose into more glycogen in the liver and muscles, and also converts it into triacylglycerols fats, which are then stored in adipose tissue.
So storing it for later, either as glycogen or fat.
Exactly. This constant balance between fuel use and storage across different tissues, the intertissue balance shown in figures 4Vog9 and 4Vog10, is precisely coordinated. And the coordinators are hormones.
Ah, the regulators we mentioned earlier.
Yes, hormones like insulin and glucagon are the primary players, along with others like cortisol, epinephrine, and norepinephrine. They act as this finely tuned communication system, ensuring fuel supply constantly adapts to our physiological state: eating, fasting, exercising, stress.
Okay, so the body works tirelessly to keep glucose levels just right, using various fuels and storage tricks. But what happens if this intricate system breaks down? Or if the energy-generating machinery itself, like that ETC turbine, falters? This sounds like it could have some really serious implications.
Precisely. And that's where the critical connection to clinical conditions comes in. The body's constant energy generation relies fundamentally on the oxidation of fuels, which, remember, consumes oxygen and produces ATP. When this process is compromised, maybe due to oxygen deprivation, maybe metabolic defects, the consequences can be severe, even life-threatening. The whole process of energy generation via fuel oxidation and oxygen consumption is called cellular respiration. Figures IV.11 and IV.2 outline this. And it's staggering. Over 90% of the oxygen you inhale is dedicated solely to this process.
90%. Just for making ATP.
Primarily, yes. Cellular respiration happens in two main phases. Phase 1 is the oxidation of fuels. Molecules like glucose, fatty acids, ketone bodies, amino acids are systematically broken down. This generates those reduced coenzymes we mentioned, NADH and FADH₂. These are like the electron-carrying taxis.
Right, the taxis.
And these various fuels often converge to form a key 2-carbon molecule called acetyl-CoA. Acetyl-CoA then enters the TCA cycle for further oxidation. Then comes phase 2, oxidative phosphorylation. This is where the real ATP powerhouse resides. The energy captured in those electron taxis, NADH and FADH₂, is used here. The electrons are passed down the electron transport chain, ETC, which is a series of protein complexes embedded in the inner mitochondrial membrane. Think of it like a molecular bucket brigade for electrons.
Passing electrons down the line.
Yes, and as electrons move down the chain, energy is released and used to pump protons across the membrane, creating an electrochemical potential like building up water pressure behind a dam. This potential energy then drives the synthesis of ATP by a remarkable enzyme complex called ATP synthase, sometimes called F0F1 ATPase. It acts like the turbine in the dam, using the flow of protons back across the membrane to physically rotate and synthesize ATP.
So the mitochondria are where this big energy payoff happens.
Yeah. Exactly. Mitochondria are the primary site for oxidative phosphorylation. The ATP generated there is then transported out to the cytosol to power everything the cell needs to do. It's an absolutely incredible feat of molecular engineering.
It really sounds like it. Now, do different fuels give us the same amount of ATP? You mentioned fatty acids earlier.
Good question. No, they don't.
Yeah.
Different fuels yield different amounts of energy, and the body uses this strategically. Fatty acids are incredibly energy dense. They're stored as triacylglycerols in adipose tissue. When needed, they're released into the blood and oxidized in the mitochondria via a process called oxidation. This chops them down into acetyl-CoA units, which then feed into the TCA cycle. Think of fats as the body's long-term, high-capacity fuel storage.
More bang for your buck, metabolically speaking.
Definitely, per gram. Amino acids, from dietary protein or breakdown of body proteins, can also be used for fuel. They can be converted to acetyl-CoA, or intermediates of the TCA cycle, or even used to make glucose via gluconeogenesis, especially during prolonged fasting. However, oxidizing amino acids generates ammonia, which is toxic. So the liver efficiently converts this ammonia into urea, which is then excreted in urine.
The necessary cleanup step.
A vital one. And glucose, as we discussed, is central. One molecule of glucose yields only about 2 net ATP through anaerobic glycolysis. But under aerobic conditions, going through the TCA cycle and ETC, it yields significantly, typically around 30 to 32 ATP molecules per glucose. Much more efficient.
A huge difference when oxygen is available.
A massive difference. Now, when problems occur in fuel oxidation, the consequences can be pathological, as Figure 4.33 illustrates. Problems can arise from, say, a lack of a required product or a substrate, maybe an enzyme deficiency blocking a pathway, or it could be due to inadequate ATP production. This might stem from oxidative phosphorylation defects, maybe caused by toxins like cyanide, which famously blocks the ETC, or, more commonly, oxygen deprivation, hypoxia.
Like in a heart attack.
Exactly. Myocardial ischemia leading to a heart attack is a prime example. The lack of oxygen forces heart muscle cells into anaerobic glycolysis. They produce some ATP, but way less, and they also produce lactate. This buildup of lactic acid contributes to cell damage and the pain associated with a heart attack.
That burning feeling during intense exercise, is that related?
Yes, that muscle burn is often due to lactate buildup from temporary anaerobic conditions in heavily working muscles. Other metabolic disorders can also compromise energy generation. Hyperthyroidism, for instance, cranks up the metabolic rate, causing excessive heat generation and energy wastage. Anorexia nervosa involves severely reduced fuel intake. Even vitamin deficiencies, like thiamine deficiency, can impact key enzymes in fuel metabolism. And subtle mutations in mitochondrial DNA can directly impair oxidative metabolism.
So many potential points of failure.
It's a complex system. And understanding the clinical context often involves knowing some basic medical terminology, prefixes, and suffixes.
Oh, yeah, you mentioned that. Can you give a quick recap?
Sure. Things like NE without edi for blood, hyper for excessive, hypo for deficient, achosis for an abnormal or disease state, and urea related to urine. So when you hear lactic acidosis, you know it's an abnormal state involving excess lactate, or ketoacidosis, an abnormal state with excess ketone bodies, often seen in uncontrolled diabetes. Knowing these helps decipher clinical terms, the regulators, hormones, and metabolic homeostasis, based on Chapter 19.
All right, we've sketched out the energy pathways, the fuels, the ATP currency, but you mentioned hormones as the conductors. If the body's running all these complex pathways, who is directing this biological orchestra? This next section, diving into hormone regulation, sounds really fascinating. How does the body manage this dynamic equilibrium?
Indeed. Chapter 19 really focuses on this concept of metabolic homeostasis. It's not a static state, but a precise, active, dynamic regulation, constantly balancing fuel availability with the needs of different tissues, like a very sophisticated thermostat for your body's energy. This balance is achieved through a complex intertissue communication system. It involves sensing the levels of circulating nutrients like glucose, crucial hormonal signals, which are our main focus here, and input from the nervous system too. Our cells have this continuous, relentless demand for ATP. So maintaining a constant supply of fuels in the blood is absolutely critical for survival and just basic daily function. Figure 19.1 gives a nice visual overview of this balance.
And you said blood glucose is a key part of that balance.
Absolutely key. Blood glucose levels are kept within that very narrow range we discuss, typically 80 to 100 milligrams per deciliter, or about 5 millimolar, which is remarkable given how much our carbohydrate intake can vary feast or famine, sometimes within hours. So the body must constantly adjust its internal systems. This constant balancing act is glucose homeostasis. And different tissues have different fuel preferences, adding another layer of complexity.
Right, like the brain needing glucose.
Exactly. The brain primarily uses glucose, needing about 150 grams a day. The liver is incredibly versatile. It can use and produce glucose, fatty acids, ketone bodies. It's the central metabolic hub. Adipose tissue stores fatty acids as triacylglycerols, but releases them as an alternative fuel when glucose is low. And skeletal muscle stores its own glycogen for quick use and can also burn fatty acids and glucose. The interplay between these tissues, orchestrated by hormones, maintains that crucial glucose stability.
That tight range for blood glucose is really striking. Which brings us back to the big players, insulin and glucagon. We hear those names constantly, especially regarding diabetes. How do they really work together or maybe against each other to keep us balanced?
They're essentially the two primary commanders, operating in a dynamic opposition to regulate blood glucose. Insulin is the major anabolic hormone. Anabolic means building up, so insulin promotes fuel storage.
Insulin stores energy.
Yes. Glucagon is its main counter-regulatory counterpart. It does the opposite. It mobilizes stored fuels.
Glucagon releases energy.
Precisely. Think of insulin saying, store energy for later, and glucagon saying, release energy now. This constant push and pull is critical. Let's look at insulin first, referencing Table 19.1 and Figure 19.2a. Insulin is released from specialized cells in the pancreas, called beta cells, located in clusters called the islets of Langerhans. Its release is primarily triggered by rising blood glucose levels, usually after you eat carbohydrates. Its main function is to lower blood glucose. It achieves this by promoting glucose uptake and use in tissues like muscle and adipose tissue, stimulating the storage of glucose as glycogen in the liver and muscles, and promoting the conversion of excess glucose to fat in adipose tissue. It also encourages protein synthesis and general cell growth.
So insulin is the fed state hormone telling cells to take up and store nutrients.
That's a perfect way to think about it. Now, glucagon, also in Table 19.1 and Figure 19.2b, is released from different pancreatic cells, the alpha cells. Its release is triggered mainly by falling blood glucose levels, like during fasting or between meals. Its job is to mobilize stored fuels to increase blood glucose. It primarily acts on the liver, promoting glycogenolysis, the breakdown of stored liver glycogen, and gluconeogenesis, the synthesis of new glucose from amino acids and lactate. Glucagon also activates the release of fatty acids from adipose tissue, providing an alternative fuel source for many tissues, which helps spare the precious glucose for the brain.
So glucagon is the fasting state hormone, ensuring fuel is available when we're not eating. A constant tug of war.
Vital tug of war, yes.
Yeah. But are insulin and glucagon the only players in this regulation game? What other hormones jump in, especially during, say, stress or exercise, to influence our metabolism?
They're definitely the main players for day-to-day, meal-to-meal glucose regulation. But several other hormones play absolutely key roles, particularly when the body faces stress or increased energy demands. You can see these listed in Table 19.1 and Figure 19.9. A major one is epinephrine, commonly known as adrenaline, our classic fight-or-flight hormone.
Right, the adrenaline rush.
Exactly. It's released from the adrenal medulla part of the adrenal gland during acute stress, intense exercise, or even prolonged fasting. Epinephrine acts very rapidly to mobilize fuels. It stimulates glucose production from glycogen in both muscle and liver, and strongly promotes fatty acid release from adipose tissue. Its actions largely oppose insulin, ensuring immediate energy availability for demanding situations.
Preparing the body for action.
Instantly. Then there's cortisol. This is a different type of hormone, a glucocorticoid, released from another part of the adrenal gland, the adrenal cortex, primarily during stress, both acute and chronic. Cortisol works over a longer time scale than epinephrine. It helps the body adapt to sustained changes in energy requirements. It stimulates the breakdown of muscle protein to provide amino acids for gluconeogenesis in the liver, helping maintain blood glucose during prolonged stress or fasting. It also promotes fatty acid release from adipose tissue.
So cortisol helps manage long-term stress energy needs?
Yes, it helps ensure fuel availability over the longer haul during stressful periods. Like epinephrine, its actions generally oppose insulin. Finally, norepinephrine has effects very similar to epinephrine. It acts both as a hormone released from the adrenal medulla and, significantly, as a neurotransmitter released by nerve endings. So it's involved in both hormonal and direct neural signals for metabolic control.
So it's a complex network: insulin, glucagon, epinephrine, cortisol, norepinephrine, all working together, sometimes opposing each other, to keep fuel supply matched to demand.
Precisely. It's an intricate hormonal symphony that keeps our energy systems humming along adapting constantly to whatever life throws at us.
This hormonal orchestration is really something. So these hormones are sending messages, telling cells to store or release fuel. But how do the cells actually hear these messages? What's the mechanism? It sounds like a cellular secret language.
You've hit on a core concept: signal transduction. That's the term for this elaborate cellular communication system. It's a chain of events that transmit the signal from outside the cell to inside, triggering a specific response. Now, polypeptide hormones like insulin and glucagon are large and water-soluble, so they can't just slip through the cell membrane. They need to bind to specific receptors on the cell surface, acting like a key fitting into a lock.
Okay, so the receptor's on the outside.
Yes. Let's look at how they're made first. Insulin is synthesized in those pancreatic beta cells as a precursor molecule called proinsulin. Figure 19.10 shows this nicely. Proinsulin is a single polypeptide chain with three parts: A, B, and C peptide regions. Inside the cell, in the Golgi apparatus and secretory vesicles, proinsulin is precisely cut by enzymes, a process called proteolytic cleavage. This releases the active insulin, which consists of the A and B chains linked together by disulfide bonds, and also releases the C peptide separately.
So the C-peptide gets released too. Is it important?
It's released in equal amounts to insulin, which is actually useful clinically. Measuring C-peptide levels can give doctors an idea of how much insulin the patient's own pancreas is producing, even if they are taking insulin injections. Both insulin and C-peptide are stored in vesicles and released together into the blood by exocytosis. Interestingly, zinc ions help insulin crystallize, which aids in its storage.
Okay, and what triggers that release? You said high glucose.
Primarily high glucose, yes. The regulation is quite sophisticated, as shown in Figure 19.1 and Table 19.2. Beta cells sense glucose via a specific transporter, GLUT2. Higher glucose leads to increased metabolism inside the beta cell, producing more ATP. This rise in ATP closes specific potassium channels, KATP channels, in the cell membrane. Closing these channels changes the cell's electrical potential, opening calcium channels. Calcium ions rush into the cell, and this influx of calcium is the direct trigger for the vesicles containing insulin and C-peptide to fuse with the cell membrane and release their contents.
Wow, that's a detailed chain reaction just to release insulin.
It is. Other things can stimulate release too, like certain amino acids, signals for the vagus nerve, and gut hormones like GIP and GLP-1 that are released after a meal, sort of anticipating the rise in glucose.
And what inhibits insulin release?
Epinephrine is a major inhibitor. During stress, epinephrine overrides the glucose signal and suppresses insulin release, ensuring glucose remains available for the brain and muscles. Now, glucagon, it's synthesized in the pancreatic alpha cells, also starting as a precursor. Proglucagon, which is then cleaved to the active 29 amino acid peptide. Glucagon has a very short half-life in the blood, only about 3 to 5 minutes, which allows for rapid adjustments in fuel mobilization.
We're on quick off.
Exactly. Its regulation is outlined in Table 19.3 and Figure 19.1. Glucagon release is stimulated primarily by low glucose levels, signaling the need to raise blood sugar. Certain amino acids also stimulate its release, which is interesting, especially after a high-protein, low-carb meal. This prevents the insulin release due to amino acids from causing hypoglycemia.
Ah, protective mechanism.
Yes. And glucagon release is inhibited by high glucose and also directly by insulin itself. There's communication between the alpha and beta cells in the pancreas. Stress hormones like epinephrine actually stimulate glucagon release, further boosting fuel mobilization during fight or flight.
Okay, so that's how they're made and released. Now, the signal transduction, how do they actually act on target cells?
Right, the mechanism of action. Since insulin, glucagon, and epinephrine can't enter the cell, they bind to those surface receptors. Insulin binds to a specific type called a tyrosine kinase receptor. Binding causes the receptor itself to become activated through autophosphorylation. It adds phosphate groups to itself. This activated receptor then phosphorylates other proteins inside the cell, like one called IRS1, insulin receptor substrate 1. This kicks off a complex cascade of further phosphorylation events, leading to insulin's various effects. It reverses the effects of glucagon, like turning off glycogen breakdown. It activates enzymes involved in fuel storage, like glycogen synthesis. It promotes the synthesis of certain proteins, acts as a growth factor, and importantly, stimulates the transport of glucose and amino acids into cells.
How does it stimulate transport?
A key mechanism, especially in muscle and fat cells, is causing glucose transporters called GLUT4, which normally reside inside the cell, to move to the cell surface. This dramatically increases the cell's ability to take up glucose from the blood.
So insulin basically puts more glucose stores on the cell surface.
That's a great way to visualize it. Now, glucagon binds to a different type of receptor, a G-protein coupled receptor, GPCR. Binding activates an enzyme called adenylate cyclase inside the cell membrane. Adenylate cyclase then produces a small molecule called cyclic AMP, CMP, from ATP. CMP acts as a crucial second messenger.
A second messenger, meaning it relays the signal inside.
Exactly. The hormone is the first messenger outside. CMP is the second messenger inside. CMP then activates another enzyme called protein kinase A, PKA. PKA is the workhorse here. It phosphorylates various key regulatory enzymes within the cell. This phosphorylation acts like an on-off switch. Typically, for glucagon signaling, PKA phosphorylation activates enzymes involved in fuel mobilization, like glycogen breakdown, and inhibits enzymes involved in fuel storage, like glycogen synthesis.
So phosphorylation can turn things on or off depending on the enzyme.
Precisely. It depends entirely on which enzyme PKA targets. And the signal is turned off quickly, too, because KRMP is rapidly broken down by an enzyme called phosphodiesterase.
Ah, and you mentioned caffeine inhibits that enzyme.
Yes, caffeine and related methylxanthines inhibit phosphodiesterase, which means CAMP sticks around longer, prolonging the effects of hormones like glucagon and epinephrine. That contributes to caffeine's stimulant effects.
Interesting. What about epinephrine and norepinephrine?
The catecholamines epinephrine and norepinephrine act through similar mechanisms. They bind to various adrenergic receptors on cell surfaces, which are also GPCRs. There are different subtypes, alpha and beta. Beta-adrenergic receptors, like beta-1, beta-2, beta-3, primarily activate the same adenylate cyclase GAM-PPKA pathway as glucagon, leading to fuel mobilization, increased heart rate, etc. Alpha-1 adrenergic receptors work through a different pathway involving calcium ions as second messengers, also leading to effects like glycogen breakdown in the liver. Figure 19.3 shows some of this. Together, these hormones prepare the body for immediate action.
Okay, so most of these act via surface receptors and second messengers. What about hormones like cortisol?
Good question. Steroid hormones like cortisol and also thyroid hormones, T3, T4, are different. They are lipid-soluble, so they can diffuse directly across the cell membrane. Inside the cell, they bind to some specific intracellular receptors, either in the cytoplasm or the nucleus. This hormone receptor complex then moves into the nucleus and binds directly to specific DNA sequences near target genes. This binding directly changes the rate at which those genes are transcribed into RNA and ultimately translated into proteins. So these hormones work by directly altering gene expression.
Which sounds like a slower but maybe more sustained effect.
Exactly. It takes longer to see the effects because it involves changes in protein synthesis, but the effects can last much longer than those mediated by second messengers like CANapX.
This detailed look at signal transduction is incredible. It really pulls back the curtain on how precise these systems are, but it also vividly explains why things can go so dramatically wrong when these systems are out of whack. Can you give us some compelling clinical examples that really bring these biochemical concepts to life?
Absolutely.
Yeah.
This is where the rubber meets the road, understanding how these intricate regulatory systems manifest clinically when they are impaired. Table 19.4 in the source material provides several powerful examples. Let's start with Deborah S. Imagine her situation. Presenting with obesity and persistently high blood glucose. She's a classic example of type 2 diabetes mellitus. The key issue for her is insulin resistance.
Meaning her body makes insulin, but her cells don't respond properly.
Exactly. Her tissues, particularly muscle and adipose tissue, are resistant. They don't respond normally to insulin signal, even though her pancreas might be producing normal or even high levels of insulin trying to compensate. It's like her cells are ignoring insulin's knock at the door. This resistance leads to persistent hyperglycemia, high blood sugar. Insulin resistance is very strongly linked to obesity, and if it's not managed, it can lead to severe long-term complications.
Like damage to eyes, nerves, kidneys.
Precisely. Microvascular complications like diabetic retinopathy, eye damage, neuropathy, nerve damage, and nephropathy, kidney disease. And also macrovascular problems increased risk of heart attacks and strokes due to damage to larger blood vessels. Her elevated HbA1c level of 9.5% is a critical sign. HbA1c reflects average blood glucose over the past few months, so 9.5% indicates very poor long-term glucose control.
Okay, that's type 2. What about type 1? You mentioned Diane.
Diane represents type 1 diabetes mellitus. This is fundamentally different. It's an autoimmune disorder. The body's own immune system mistakenly attacks and destroys the insulin-producing beta cells in the pancreas. This leads to an absolute deficiency of insulin. Her body simply cannot make it. Patients like Diane require lifelong administration of exogenous insulin, usually through injections, or an insulin pump, just to survive and manage their blood glucose. Without it, they would rapidly develop severe metabolic derangement, ketoacidosis, and coma.
A complete lack of insulin versus resistance to it.
Yeah.
Very different.
Very different causes and initial treatments. Then consider Connie C. She experienced worrying episodes of fatigue, confusion, blurred vision. These are neuroglycopenic symptoms.
Neuroglycopenic, meaning brain-related low glucose symptoms.
Exactly. Her brain wasn't getting enough glucose, its primary fuel, leading to cognitive impairment and neurological symptoms. Her episodes were also often accompanied by adrenergic symptoms, palpitations, anxiety, sweating.
That's the fight or flight response kicking in?
Yes, her body releasing counter-regulatory hormones like epinephrine, desperately trying to raise her dangerously low blood glucose. Connie was diagnosed with an insulinoma.
A tumor.
A tumor of the pancreatic beta cells, yes. But this tumor secretes massive amounts of insulin inappropriately, completely unregulated by blood glucose levels. This constant insulin flooding causes severe, recurrent hypoglycemia. It really highlights the dangers of too much insulin activity.
So we need just the right amount, perfectly regulated.
Precisely. Genetics also plays a role in some forms of diabetes. Maturity-onset diabetes of the young, MODY, is a group of rare inherited forms of diabetes. Specifically, MODY type 2 is caused by a mutation in the glucokinase enzyme within the beta cells. Glucokinase acts as the glucose sensor. This mutation results in a higher glyco for glucose.
Higher KMS, meaning the sensor is less sensitive, needs more glucose to react.
Exactly. It's a bit numb to glucose. It requires higher glucose levels than normal to trigger insulin release. This leads to impaired insulin secretion and mild chronic hyperglycemia. Often, it's manageable without insulin injections. Another, even rarer condition is neonatal diabetes. Here, newborns develop diabetes within the first few months of life. It's often caused by mutations in that KATP channel subunit we talked about earlier, the potassium channel involved in insulin release, often the KCNJ11 gene. This mutation prevents glucose from triggering insulin release, leading to severe hyperglycemia right from birth.
Understanding these channels must have led to treatments, right?
Absolutely. The drug application is clear. Sulfonylurea compounds, like glipizide, are a class of drugs used to treat certain types of diabetes, including some forms of type 2 and MODY. These drugs work by specifically targeting and closing those KTP channels in the beta cells. By closing the channels, they mimic the effect of high ATP, leading to calcium influx and stimulating insulin release, even if the glucose sensing mechanism is impaired.
So the drug bypasses the faulty sensor and directly triggers insulin release.
In essence, yes. These clinical examples truly underscore how critical the precise regulation of glucose metabolism and hormone action is. Even small disruptions in these intricate pathways can lead to a wide spectrum of complex, often life-altering diseases.
3.
The engine. Cellular bioenergetics and ATP, based on Chapter 20.
We've explored the fuel management system, the hormonal regulators, but let's get down to the absolute basics now. How do we actually get useful work out of all this? Chapter 20 delves into cellular bioenergetics, the real nitty-gritty of energy transformation that powers literally everything in our cells.
This chapter really does bring it all together, focusing on bioenergetics, the study of how cells transform and utilize energy. And the core principle, the absolute engine driving it all, is the ATP-ADP cycle. You can picture this in Figure 20.1. It's a continuous cycle. Fuels, carbs, fats, proteins contain chemical bond energy. When these fuels are oxidized, broken down, that released energy is captured to form ATP from ADP and phosphate. Then, ADP is hydrolyzed. That high-energy phosphate bond is broken, releasing energy back to power all sorts of cellular work: mechanical, transport, biochemical synthesis.
So it's constantly being built up using fuel energy, then broken down to release energy for work.
Exactly. It's the immediate, readily spendable energy currency, like a rechargeable battery that's constantly being discharged to power things and then immediately recharged by the cellular power plants. This cycle is the very engine of life.
And you mentioned ATP's high-energy bonds. That's where the power comes from.
Yes, specifically the phosphoanhydride bonds linking the second and third phosphate groups. Breaking these bonds releases a significant amount of energy, about 7.3 kilocalories per mole under standard conditions. This released energy is then harnessed very efficiently by the cell. Maintaining a constant level of ATP within the cell, ATP homeostasis, is therefore critical. The rates of fuel burning and ATP usage are tightly coupled and regulated to ensure this balance. Disruptions here, as we've touched on, can lead to disease, obesity, hyperthyroidism, heart attacks. All involve disruptions in energy balance or ATP production utilization.
Okay, so ATP is the currency, But how do we measure the actual value of a biochemical reaction? How much energy does it yield, or how much does it cost? That sounds like thermodynamics.
It is exactly thermodynamics. That's where the concept of Gibbs free energy, or ADG, comes in. Table 20.1 summarizes this. AG represents the change in free energy during a reaction. Essentially, it tells us two key things: whether a reaction can happen spontaneously and the maximum amount of useful work that can be obtained from it.
Spontaneous or not, how does AG tell us that?
By its sign. A negative phase means the reaction is exergonic. It releases energy and can proceed spontaneously without needing energy input like a ball rolling downhill. A positive phase means the reaction is endergonic.
Sure.
It requires an input of energy to proceed. It's thermodynamically unfavorable on its own, like pushing that ball uphill.
Okay, exergonic releases energy, endergonic requires energy. Where does ATP fit in?
ATP hydrolysis, breaking that high-energy bond to form ADP and phosphate, is highly exergonic. It has a large negative U. This means it releases a significant amount of energy, and this released energy is the key. It can be coupled to drive otherwise unfavorable endergonic reactions. This coupling is the essence of how ATP functions as the cell's energy currency. It pays the energy cost for reactions that wouldn't happen on their own.
Can you give me an example of coupling?
Sure. Remember hexokinase phosphorylating glucose to glucose 6-phosphate? That reaction, glucose plus pi, glucose 6p, is actually endergonic, with a positive voltage of about plus 3.3 kilocalmol. It needs energy. But ATP hydrolysis, ATP, AEP plus pi, is highly exergonic, with AGs of about negative 7.3 kilocalmols. When the cell couples these two reactions together, glucose plus ATP, glucose 6p plus ADP, the overall free energy change becomes negative, around negative 4.0 kilomol. So the energy released by breaking ATP's bond pays for adding the phosphate to glucose, making the whole process favorable. Table 20.3 illustrates this coupling.
So ATP makes the impossible possible, biochemically speaking.
In many cases, yes. It's also important to remember that the actual G inside a cell depends not just on the standard GC value, but also on the actual concentrations of the reactants and products. Cells manipulate these concentrations to keep reactions flowing in the right direction.
Is ATP the only high-energy molecule cells use?
Primarily, yes, it's the main currency. But there are other important activated intermediates with high-energy bonds used for specific purposes. Think of them as specialized forms of currency. Figure 20.7 touches on some. For example, uridine triphosphate, UTP, is primarily used in glycogen synthesis. Cytidine triphosphate, CTP, is crucial for lipid synthesis. And guanosine triphosphate, GTP, is vital for protein synthesis and also plays roles in signal transduction.
So specialized energy carriers for specific big jobs.
Exactly. It allows for finer control over these major biosynthetic pathways. There's also creatine phosphate. This molecule contains a very high-energy phosphate bond and acts as an immediate energy reserve, especially in cells with high fluctuating energy demands, like muscle cells and neurons. During sudden bursts of activity, when ATP is used up rapidly, creatine phosphate can quickly donate its phosphate group back to ADP, regenerating ATP almost instantly. It acts like a small, rapidly accessible backup battery.
Buffering the ATP levels during intense activity.
Precisely. It buys time for the slower main ATP-generating pathways like glycolysis and oxidative phosphorylation to ramp up.
So beyond just fueling individual chemical reactions, ATP performs actual cellular work. What are the main categories of jobs it does inside our cells, the tangible outcomes of spending this energy currency?
ATP powers three major categories of cellular work. It's incredibly versatile. First, there's mechanical work. The most obvious example is muscle contraction, as shown in figure 20.4. ATP hydrolysis drives conformational changes in proteins like myosin, causing them to walk along actin filaments, making the muscle fibers slide past each other and contract. Every heartbeat, every step you take is powered by countless ATP molecules being broken down.
Movement itself.
Yes. Second, there's transport work. Many essential processes rely on moving ions and molecules across cell membranes against their concentration gradients, moving them from low concentration to high concentration. This requires energy, provided by ATP hydrolysis. Specific protein pumps, called ATPases, like the famous Na plus K plus ATPase, the sodium-potassium pump, use ATP to actively transport ions. This is vital for maintaining cell volume, generating nerve impulses, absorbing nutrients in the gut. Countless functions depend on these ATP-powered pumps.
Maintaining gradients and enabling signaling.
Absolutely. Third, there's biochemical work. This broad category encompasses all the anabolic building up reactions, synthesizing large complex molecules like DNA, RNA, proteins, glycogen, fats, all require energy input, primarily from ATP. It fuels the construction processes that allow cells to grow, repair themselves, and replicate. ATP also powers detoxification reactions, where enzymes modify toxic compounds to make them less harmful and easier to excrete.
Building, repairing, detoxifying, it really does everything.
It's the fundamental power source for nearly all cellular activities.
And all this energy generation and use, it must create some byproducts, right? Like heat. You mentioned hyperthyroidism generating heat. And what about situations where oxygen runs low, like during really strenuous exercise or, more critically, during a heart attack?
Absolutely. Heat production, or thermogenesis, is a natural and essential consequence of metabolism. The laws of thermodynamics dictate that no energy conversion is 100% efficient. As chemical energy from fuels is converted into the chemical energy of ATP, some energy is inevitably lost as heat. But this heat isn't wasted. It's crucial for maintaining our body temperature within the narrow range needed for our enzymes to function optimally. Our basal metabolic rate, BMR, largely reflects the energy spent on basic functions and this necessary heat production. Things like shivering, involuntary muscle contractions, and non-shivering thermogenesis, specialized metabolic processes, are ways the body actively generates more heat when needed.
So some heat is normal and necessary.
Yes. Now, fuel oxidation is the primary source of the energy captured in ATP, generating those reduced coenzymes, NADH and FADH₂. It's worth reiterating that NADPH from the pentose phosphate pathway is different. It's used for biosynthesis and antioxidant defense, not directly for ATP synthesis via the electron transport chain. These processes are all oxidation reduction reactions. Fuels get oxidized, lose electrons, and ultimately oxygen gets reduced, gains electrons in the ETC. The tendency of a compound to gain or lose electrons is measured by its reduction potential. Electrons naturally flow from compounds with lower, more negative reduction potential to those with a higher, more positive potential, like flowing downhill. NADH and FADH₂ have relatively low reduction potentials, while oxygen has a very high one. So electrons flow spontaneously down the ETC from NADH-FAD₂ towards oxygen, releasing energy along the way.
Oxygen is the ultimate electron acceptor.
In aerobic respiration, yes. That's why it's essential. Regarding caloric values, we touched on this. Fatty acids yield about 9 kilograms, while glucose and proteins yield about 4 kilocalogram. Fats are more reduced, have more CH bonds, and less oxygenated initially, hence more energy is released when they are fully oxidized.
Okay, now about limited oxygen conditions.
Right. The body has an important backup, anaerobic glycolysis, shown in figure 20.11. As we discussed, when oxygen is scarce, like in intensely working muscle or red blood cells without mitochondria, cells can still generate some ATP by breaking down glucose only as far as lactate. It's much less efficient than aerobic respiration, yielding only 2 net ATP per glucose compared to 30-32. But it's fast and doesn't require oxygen. It's a vital survival mechanism in certain situations or cell types, but prolonged reliance on it isn't sustainable for most tissues due to lower ATP yield and lactate buildup.
And you mentioned enzymes using oxygen differently, oxidases and oxygenases.
Yes, figure 20.12 distinguishes these. Think of oxygen's role. Oxidases use O2 primarily as an electron acceptor, typically reducing it to water, H2O, or hydrogen peroxide, H2O2. The final enzyme in the ETC, cytochrome C oxidase, is a prime example. It transfers electrons to oxygen to make water. Oxygenases, on the other hand, directly incorporate one or both atoms from O2 into the substrate molecule. They use oxygen as a reactant in a chemical transformation. These are important in many biosynthetic pathways, like making certain amino acids or hormones, and also in detoxification, where adding oxygen atoms can make compounds easier to modify and excrete.
This all really ties into our overall energy balance, doesn't it? You mentioned the muscle burn from lactate during exercise. That's a direct feeling related to anaerobic metabolism, and what happens when that critical energy balance is severely disrupted, especially in life-threatening situations like a heart attack.
That's an excellent point about feeling the biochemistry. It highlights the importance of balance. Our total energy expenditure is directly linked to our oxygen consumption because aerobic metabolism is our main energy source. The body constantly strives for energy balance, where energy intake matches energy expenditure, BMR plus activity plus thermogenesis, as shown in figure 20.13. Disruptions lead to problems. Consistently taking in more calories than you expend leads to positive energy balance and storage of the excess, primarily as fat, leading to weight gain and obesity, like the case of AutoS. Conversely, conditions like hyperthyroidism, seen in Stanley T, disrupt the balance in the other direction. Elevated thyroid hormones increase ATP usage in BMR, leading to increased heat production, weight loss despite increased appetite, and symptoms like heat intolerance and rapid heartbeat. The whole system is running too fast.
And the most critical disruption is lack of oxygen.
Yes, Hypoxia or lack of oxygen is perhaps the most critical disruption for most tissues. Consider the tragic case of Cora N., who suffered a myocardial infarction, heart attack, due to ischemic cardiomyopathy, lack of blood flow, and thus oxygen to the heart muscle. When oxygen is cut off, the electron transport chain halts because oxygen is the final electron acceptor. ATP production via oxidative phosphorylation plummets. This lack of ATP has immediate and devastating cellular consequences, as depicted in Figure 20.14.
What happens inside the cell?
Without ATP, vital ion pumps like the Na plus K plus ATPase fail. Sodium ions accumulate inside the cell, potassium leaves. This disrupts the electrical potential and leads to an influx of calcium ions through channels that shouldn't be open. The increased intracellular sodium and calcium cause the cell to swell up with water. Mitochondria become damaged and dysfunctional. Eventually, the cell's structural integrity breaks down, leading to cell death, either through necrosis, uncontrolled bursting, or apoptosis, programmed cell death.
A cascade of failure triggered by a lack of ATP.
A rapid and devastating cascade. Now, the body does have some adaptive responses to low oxygen. Hypoxia-inducible factors, HIFs, are proteins that become active under hypoxic conditions. These HIFs act as transcription factors, turning on genes that help the cell adapt, for example, genes involved in making more red blood cells via erythropoietin to improve oxygen delivery, or genes for enzymes involved in anaerobic glycolysis. But these adaptations have limits, and severe or prolonged hypoxia remains a major cause of tissue damage and death in many diseases. It truly underscores our fundamental dependence on a constant supply of oxygen and ATP. The journey, digestion, absorption, and transport of carbohydrates, based on Chapter 21.
Okay, so we've charted the intricate pathways of how the body uses carbs for energy, how hormones regulate that use, and how the cellular engine, ATP, actually works. Cool, hold on, before any of that can happen, the carbohydrates we eat have to actually get into our bodies from our digestive tract. They can't just magically appear in our cells. So let's trace that entire journey right from the plate, based on Chapter 21.
Exactly. This chapter focuses on that essential first step, digestion and absorption. It's fundamental because the vast majority of carbohydrates we eat are not in a form our cells can directly use. We eat complex polysaccharides like starch or disaccharides like sucrose, table sugar, and lactose, milk sugar. These large molecules are way too big to be absorbed directly into our bloodstream. They need to be systematically broken down to their simplest units, the monosaccharides, primarily glucose, fructose, and galactose. This breakdown process is digestion.
And carbs are a big part of our diet, right?
A very significant part.
Yeah.
In the typical American diet, they make up about 40–45% of total calories. So efficient digestion and absorption are crucial. Let's look at the main types we eat. Starches, the polysaccharides, are the big ones. They come in two forms. Amylose, which is a straight chain of glucose units linked by 1,4,4 glycosidic bonds, and amylopectin, which is similar but has branches created by I-146 glycosidic bonds. You find these in grains, potatoes, rice, bread, pasta.
Staple foods for many people.
Absolutely. Then we have the common disaccharides. Lactose in milk is glucose linked to galactose via a 1,4 bond. Sucrose, table sugar, also in fruits, some vegetables, is glucose linked to fructose via an ubiquitous bond. And maltose, which is mainly a product of starch digestion, consists of two glucose units linked by an ab-1 bond. We should also mention dietary fiber. These are also polysaccharides like cellulose or pectin, but they have bond types, like the 1,4 bonds in cellulose, that our human digestive enzymes simply cannot break down.
Okay, so starch, sucrose, lactose need breaking down. Fiber doesn't, by us anyway.
Yep.
So step one, breaking down those large starches. Where does that process kick off?
It starts right away, in your mouth. As you chew, saliva mixes with your food, and saliva contains an enzyme called salivary amylase. This enzyme begins hydrolyzing the ab-1 glycosidic bonds within the starch molecules, breaking the long chains into shorter fragments.
So digestion starts immediately.
It does, but its action is quite limited. Salivary amylase works best at the neutral pH of the mouth, but it gets rapidly inactivated by the strong acidity of the stomach once you swallow. So, while it gives starch digestion a head start, the really significant starch breakdown happens further down, in the small intestine.
What happens there?
Once the stomach contents move into the small intestine, the pancreas releases a potent cocktail of digestive enzymes, including pancreatic amylase. This pancreatic amylase picks up where the salivary one left off, continuing to hydrolyze the O1,4 glycosidic bonds in starch and starch fragments. Figures 21.2 and 21.3 illustrate this process. Importantly, like salivary amylase, pancreatic amylase cannot break the OM16 branch points found in amylopectin, nor can it cleave the OM4 bonds right at the ends of the chains or next to a branch point.
So it breaks starch down, but not completely. What are the results?
The main products of amylase digestion are smaller oligosaccharides, short chains of glucose. These include maltose, 2 glucose units, maltotriose, 3 glucose units, and so-called I-limit dextrins. These dextrins are branched oligosaccharides containing those I1-Vela6 linkages that amylase couldn't touch. So we've gone from huge starch molecules down to smaller sugars, but mostly still not the single glucose units we need for absorption.
Okay, so the amylases handle the bulk starch breakdown, leaving us with disaccharides like sucrose and lactose from our diet, plus maltose and these limit dextrins from starch. We still need to get to individual glucose, fructose, and galactose molecules. How does that final crucial snip happen?
This is where the action moves right to the surface of the small intestine lining. The cells lining the small intestine, the enterocytes, have a specialized surface called the brush border. It's covered in tiny finger-like projections called microvilli, which massively increase the surface area for absorption. Embedded right within the membrane of this brush border are the crucial enzymes that perform the final breakdown, the disaccharadases. Think of them as molecular scissors, perfectly positioned to snip the remaining disaccharides and oligosaccharides into their constituent monosaccharides just before absorption. They are integral membrane proteins. Their active sites facing the intestinal lumen. Table 21.1 and Figure 21.4 show these key players.
What are the main disaccharadases?
There are several important ones, each with specificity. Glucoamylase continues the work on starch fragments, hydrolyzing IO104 bonds from the ends of oligosaccharides, releasing glucose. It's good at handling longer chains. Then there's a very important enzyme complex called the sucrose isomaltase complex. It's actually one large protein with 2 different active sites.
2 enzymes in one?
Essentially, yes. The sucrose site hydrolyzes sucrose, table sugar, into 1 glucose and 1 fructose molecule. The isomaltase site, also called idextrinase, is crucial because it's the only enzyme that can hydrolyze the ICO16 glycosidic bonds found in those limit dextrins from amylopectin, releasing glucose. It also hydrolyzes maltose.
So that one handles sucrose and the branch points from starch. Very important.
Absolutely essential for complete starch digestion. Another critical enzyme is lactase, technically part of the lactase-glutosylceramidase complex. Its main job is to hydrolyze lactose, milk sugar, into glucose and galactose. Lactase activity is typically highest in infants and then declines in most populations after weaning, which is the basis for lactose intolerance. And finally, there's trehalase, which hydrolyzes trehalose, a disaccharide found in things like mushrooms and insects, into 2 glucose molecules.
So these brush border enzymes are the final gatekeepers, ensuring everything is broken down to single sugars.
Exactly. They perform the final hydrolysis, releasing glucose, fructose, and galactose right at the surface where absorption can happen.
Okay, now we have the monosaccharides glucose, fructose, galactose ready to go. How do they actually get across the intestinal cell membrane and into the bloodstream to fuel the rest of the body?
This absorption step is also highly specialized, involving specific transporter proteins embedded in the enterocyte membrane. It's not just simple diffusion for most of them. Glucose and galactose share the same primary transport mechanism.
Yeah.
They are taken up into the intestinal cell via secondary active transport using a transporter called SGLT1, sodium glucose link transporter 1.
Active transport, meaning it requires energy.
Yes, indirectly. SGLT1 co-transports 1 molecule of glucose or galactose along with 2 sodium ions into the cell. It uses the energy stored in the sodium gradient, the fact that sodium concentration is much lower inside the cell than outside. This gradient is maintained by the Na plus AK plus ATPase pump, which does use ATP, located on the other side of the cell, pumping sodium back out. So SGLT1 uses the sodium gradient to actively pull glucose and galactose into the cell, even if the concentration inside is already higher than outside. This ensures very efficient absorption.
Okay, so glucose and galactose get pulled in with sodium. What about fructose?
Fructose uses a different mechanism.
Yeah.
It's transported into the cell primarily by facilitated diffusion through a transporter called GLUT5.
Facilitated diffusion, meaning no direct energy needed, but it still needs a transporter.
Exactly. Fructose moves down its concentration gradient from the higher concentration in the intestinal lumen to the lower concentration inside the cell, but it needs the GLUT5 protein to help it cross the membrane. This process doesn't require energy directly and isn't coupled to sodium.
Different mechanisms for different sugars. Interesting. And how do they get out of the intestinal cell into the blood?
Once inside the enterocyte, all 3 monosaccharides, glucose, fructose, and galactose, exit the cell across the basolateral membrane, the side facing the blood, and enter the capillaries of the portal circulation. This exit is primarily mediated by another transporter, GLUT2. GLUT2 is also a facilitated diffusion transporter, allowing the sugars to move down their concentration gradient from the high concentration inside the cell, after absorption, to the lower concentration in the blood. From there, the portal vein carries them directly to the liver, the first stop for processing newly absorbed nutrients.
So active uptake for glucosilgalactose, facilitated for fructose, then facilitated exit for all three into the blood, a very specific system.
Highly specific and efficient, designed to capture these vital fuel sources from our diet.
Now, what about dietary fiber? You said our enzymes can't digest it. It seems to bypass this whole elaborate digestion and absorption process. What happens to it then? What's its role?
That's right. Dietary fiber, composed of polysaccharides like cellulose with quinone 4 bonds and pectins, resists digestion by human enzymes. It passes largely intact through the stomach and small intestine. When it reaches the colon, or large intestine, it encounters a massive population of bacteria, our gut microbiome. These bacteria do possess the enzymes needed to break down, ferment many types of fiber.
So the bacteria digest what we can't?
To a large extent, yes. This bacterial fermentation produces several things. One byproduct is gas, primarily hydrogen, H2, carbon dioxide, CO2, and methane, CH4, which can cause flatulence. More importantly, fermentation produces short-chain fatty acids, SCFAs, such as acetate, propionate, and especially butyrate. These SCFAs are actually absorbed by the cells lining our colon and can be used by them as an energy source. Butyrate in particular is a preferred fuel for colonocytes and plays roles in maintaining gut health.
So fiber indirectly provides some fuel, at least for the colon cells.
Yes, and these SCFAs may have other systemic health benefits too, though that's an active area of research. But fiber's main roles are perhaps more mechanical and related to gut health. It adds bulk to the stool, which helps promote regular bowel movements and prevent constipation. It also helps feed the beneficial bacteria in our gut, contributing to a healthy microbiome.
So crucial for gut health, even if it's not a major direct energy source for us, like glucose.
Exactly. It plays a different but still very important role in our overall physiology.
This whole system, from mouth to absorption, sounds so finely tuned. But what happens when something goes wrong in this digestion and absorption pipeline? I'm thinking about something incredibly common, like lactose intolerance. How does that fit the picture?
Lactose intolerance is the perfect clinical example of a breakdown in this system, specifically a deficiency in one of those brush border enzymes. It's extremely common worldwide. It's characterized by those uncomfortable symptoms: abdominal pain, bloating, nausea, diarrhea, flatulence after consuming milk or dairy products containing lactose. And it occurs precisely because of low levels or absence of the lactase enzyme. Remember, lactase is needed to break lactose down into glucose and galactose for absorption.
So if you don't have enough lactase?
If you don't have enough lactase, the lactose sugar isn't broken down or absorbed in the small intestine. It travels undigested into the large intestine, the colon. Figure 21.2 helps visualize what happens next. The bacteria residing in the colon are delighted to encounter this undigested lactose. They rapidly ferment it.
Just like they ferment fiber.
Exactly. The fermentation produces gases, hydrogen, CO2, methane, leading to bloating and flatulence. It also produces lactic acid and other short-chain fatty acids. These fermentation products, along with the undigested lactose itself, are osmotically active. They draw water from the body into the colon lumen.
Pulling water into the gut.
Yes, which leads to the characteristic watery diarrhea, often called osmotic diarrhea. The gas production and intestinal distension also cause abdominal cramps and pain.
That makes sense. Are there different reasons why someone might lack lactase?
Yes, there are different types. The most common by far is non-persistent lactase activity, sometimes called adult-type hypolactasia or primary lactose intolerance. In most mammals, including a majority of humans globally, lactase enzyme production naturally declines significantly after infancy or early childhood. This is a genetically programmed downregulation. It's actually the persistence of lactase into adulthood. Common in populations with a long history of dairy farming, like Northern Europeans, that's the less common genetic trait worldwide.
So for many populations, losing lactase activity is normal.
It's the genetic norm, yes. Prevalence varies hugely, very high, in people of Asian, African, and Native American descent, much lower in Northern Europeans. Then there's congenital lactase deficiency. This is a very rare, severe inherited disorder where an infant is born with little or no ability to produce lactase at all. They develop severe diarrhea and dehydration immediately upon consuming breast milk or lactose-containing formula. The case of Nina M. in the text, the 13-month-old with severe symptoms after milk, strongly suggests this rare condition. Finally, you can have secondary lactase deficiency. This occurs when the lining of the small intestine is damaged by illness, infection, or other conditions. Things like severe gastroenteritis, celiac disease, Crohn's disease, or even malnutrition, like kwashiorkor, can temporarily or sometimes permanently damage the brush border cells, reducing lactase production.
So injury or illness can cause it too. How is it diagnosed? You mentioned gas.
The hydrogen breath test is a common diagnostic tool. Since human cells don't produce hydrogen gas, if a person drinks a lactose solution and then exhales significantly increased amounts of hydrogen, it indicates that undigested lactose reached the colon and was fermented by bacteria, producing hydrogen that got absorbed into the blood and exhaled via the lungs.
Clever, using bacterial gas production as a marker.
It is. And we see related principles in other contexts, too. Remember Deborah S., the patient with type 2 diabetes? The text mentions she used a starch blocker to help with weight loss. Starch blockers are compounds that inhibit amylase activity. By reducing starch digestion, they reduce the amount of glucose that gets absorbed, thus lowering caloric intake from starch. It's another example of targeting this digestive pathway, this time therapeutically. These examples really highlight how understanding the specifics of carbohydrate digestion and absorption is directly relevant to common human conditions and even therapeutic strategies.
Outro. Phew. Okay, we have really covered a huge amount of ground today. We've gone from the first bite of a carbohydrate, traced its breakdown and absorption, followed glucose through its complex metabolic fates, energy storage, building blocks, explored the intricate hormonal controls, and landed on ATP, the very currency of life. It's genuinely an interconnected marvel.
It truly is. We've seen how absolutely central glucose is, acting not just as that universal fuel, but also as an incredibly versatile precursor molecule. We've emphasized how critical ATP is, powering virtually every cellular function through that constant cycle of hydrolysis and regeneration. We've delved into the elegant dance of hormones like insulin and glucagon, tirelessly working to maintain that delicate blood glucose balance, adapting to the body's ever-changing needs. And we followed the practical journey of digestion, seeing how large dietary carbs are meticulously broken down and what happens clinically, as in lactose intolerance, when that process falters.
Yeah, each piece really does connect to the next. It's not just isolated facts. It forms this cohesive, logical picture of how our bodies literally run.
Exactly, it's a system.
So what does this all mean for you listening? Well, hopefully it gives you a deeper appreciation for your own body as a master of biochemical engineering. It's constantly adapting its fuel use, storage, and digestion with incredible precision. Understanding these pathways, even at this level, offers a profound insight into the complexity and elegance of human biology. Whether you're studying for an exam, trying to understand a health condition, or just, curious about how you work.
And it really underscores that this knowledge is most powerful when it's connected and applied. Understanding these fundamentals helps make sense of conditions like diabetes, the rationale behind dietary advice, the effects of exercise and stress on your energy levels. It's foundational knowledge for navigating health. We really encourage you to reflect on these connections. How does a problem in digestion, like lactose intolerance, ripple through to affect nutrient availability and maybe even long-term energy balance? How does hormonal dysregulation impact fuel partitioning across the entire body? Recognizing these interconnections, seeing the body as a unified system rather than just isolated parts, that's where the deepest understanding lies.
Thinking systemically, that's a great takeaway. Thank you so much for guiding us through that intricate journey today.
My pleasure. It's fascinating material.
And thank you all for joining us on this deep dive into carbohydrate metabolism, fuel oxidation, and bioenergetics. Keep that curiosity alive, keep asking questions, and we look forward to exploring another fascinating topic with you again soon.